on the theme of AI and biology, right? So the next talk will be on neuroscience, and we have David Freedman, who is professor in neurobiology and Neuroscience Institute at U Chicago, and having a broad background in cognitive systems and computational neuroscience with expertise in electrophysiological approaches for recording neuronal population activity in awake non-human primates trained to perform complex behavioral tasks. And this requires learning, memory, and decision making. And his research program also is a focus on artificial intelligence for studying neuroscience related questions in artificial neural networks and designing biologically inspired AI approaches. And his research, supported by NIH, NSF, DoD, and private foundations, investigates neuronal computations of higher order perceptual and cognitive functions. So following graduate and postdoctoral training at MIT and Harvard Medical School, he established his lab at University of Chicago in 2008, and he's trained numerous graduate students and postdoc researchers, and they've gone on to build their own successful careers. A number of awards. He's been recognized by the Trollent Research Award from National Academy of Sciences, the Vannevar Bush Faculty Fellowship from the Department of Defense, the NSF Career Award, and faculty fellowships from Sloan, McKnight, and Brain Research Foundation, as well as the U Chicago Faculty Award for Excellence in Graduate Teaching and Mentoring. Thank you.
All right. Well, thank you very much. It's a real pleasure to be here. This has been just a fascinating day so far. I'm grateful for being included. So this inspirational imagery that you're seeing right here was actually created from a generative AI algorithm, DALL-E, actually with many of the keywords from that introduction fed into it as a prompt. And actually that's true for the next slide as well. So we've heard so many examples today of AI being used for scientific discovery, right? It's remarkable how powerful it's been for making sense of large data sets. And I'm going to tell you some examples about that from neuroscience and from our own research, but I'm also going to tell you about an interaction between science and AI that goes in the other direction, which is actually taking inspiration from the brain and trying to understand something about what it is that makes brains so flexible, so intelligent, and so energy efficient. By the way, our brains are operating at about 20 watts of power, right? That lunch that we just had is enough to keep us going for the rest of the day. So what I'm going to tell you about is how we're using knowledge that the field of neuroscience is gaining about the brain, trying to implement that in artificial neural network models to enhance their capabilities. So the question that really drives me, that initially drove me in research, is to ask the question, how is it that we see, right? You look around this room, you look around any complex scene, and you have a remarkable ability to make sense of your surroundings. Put in a slightly more elaborate way, how does light entering our eyes evoke our visual perception, allowing us to learn, to remember, to make decisions? It's just remarkable, right? And this was a question that I encountered during my kind of undergraduate training days, and it's kind of pulled me in and captured my attention ever since. So if you're presented with a complex image, a visual scene like this one, you'll easily and almost automatically recognize many of the familiar items in a scene like this. You see people, you see buildings, familiar logos from companies and things like this. But you'll also recognize higher order information. You might recognize this as a familiar location, Times Square, New York City. But above and beyond that, you can also extract information about the broader context. You could probably make a guess about what time of day this picture was taken, maybe what the weather was like, perhaps even what year the photo was taken, based on cues that you can find in the image, right? This is remarkable, right? The brain has a remarkable ability to do this. And so, yeah, the brain has this remarkable ability to make sense of the world around us. And this is so that the brain can guide our decisions and guide our actions. And the field of neuroscience over the past decades has made quite a bit of progress in understanding the sort of basic building blocks, the basic mechanisms of vision and cognition. There's a long way to go, but I think, have a really good platform now for asking kind of higher order questions about these kinds of cognitive functions. And neuroscience-based understanding of what makes biological brains intelligent has been important for bringing the field of AI to where it is today. The example that I like to bring, because I think, or that I like to think about, because I come from a vision science background, is the remarkable strides in computer vision. Right. So if we go back about 10 years ago, and what I'm showing you here is the performance of cutting edge computer vision algorithms at different time points, 2011 through I think 2018 here, relative to human level visual recognition performance. This is on a standard image recognition benchmark. So human level image recognitions, they're around 5% error rate on this task. And in around 2016, the cutting edge computer vision systems were suddenly doing as well or even better than human subjects faced with the same task. And it's continued to improve since then. And the architecture of those artificial neural networks, these, now widespread, feed-forward convolutional neural networks, were very strongly inspired by knowledge about how the primate visual system operates, by the anatomical connections between different hierarchically connected processing stages in the primate visual system. You see areas like V1, V2, V4, PIT. These are all different regions of the visual cortex, which we know process incoming information from the retina and extract features about the images that the subject is looking at. And the rules by which information is transformed and transmitted between different processing stages has been helpful in motivating the architecture of many of the sort of leading image recognition systems. Despite this progress, right, we've seen many other examples of remarkable progress in AI. These systems still struggle to make good decisions in complex real-world context, right? Imagine placing an AI-powered agent, like an AI-powered robot or something, in the middle of Times Square with some kind of abstract, goal that you might instruct the robot, like, please go, pick up groceries, pick up dinner, and then, see if you can get good tickets for the Mamma Mia show tonight, or something like that. I don't think any of us would have high hopes for this robot coming back with all of these items, successfully checked off the list. And although any one of these sub goals, right, motor control or navigation, I think we can assume that we could probably get, an agent to solve each one of these problems, but to stitch them all together, allowing for sort of flexible behavior that can adapt to the moment-to-moment challenges that this agent, would encounter in this scene. That's too much for modern AI systems, right? But it's not too much for us, or even non-human animals can actually do pretty well at navigating around complex environments. So, what I'll pose to you is that neuroscience will continue to inspire future AI breakthroughs, and in turn, AI is also accelerating neuroscience research, not just by helping us analyze our data, but actually by actually themselves becoming better models for how the brain actually works, right? Allowing us to test some of our hypotheses in artificial networks that are trained on complex behavioral tasks. And I'll give you some examples about that today. Okay, so a bit about me. I went off to college when I was 18 years old, thinking I was going to study electrical engineering or computer science or physics. And that was really my plan. I started out that way, but I got completely sidetracked and derailed and pulled into experimental neuroscience of vision and cognition because I got so excited about the promise for making some progress here, and because I'd never been exposed to, in high school or something, the level of progress that had already been made, back already in the 90s, understanding circuits underlying these processes. And so I'm really fortunate to be here at the University of Chicago, where I've been for 15 years running a laboratory aimed at understanding mechanisms of vision and cognition with my laboratory. But inspired by my computer science and sort of early AI interests, my lab at U Chicago, in addition to experiments, we're increasingly working at the sort of interface between experimental neuroscience, doing experiments, collecting data from the brain, but then also thinking about those same questions from an AI standpoint as well. I'm also really happy to be co-directing the new Schmidt AI and Science Postdoctoral Fellowship Program here at University of Chicago, along with my co-directors, Becca Willett and Josh Freeman, who are both here, and a number of our fellows are also here. I see them all in the back. So it's been a really exciting program so far, and it's been fun to be a part of it. Okay, so this is a group out here in the audience from representing so many different fields. So I thought it'd be useful to give just a brief primer on how it is that the brain actually interprets signals from the outside sensory world coming in through the visual system. So imagine being presented with an everyday object, right? A coffee cup is presented to you. And what do you have to do? You have to figure out what to do next. So what happens? Well, light from lighting sources sends photons, some of which will reflect off of the coffee cup. And a few of those photons will bounce off the coffee cup at just the right angle so that photons will enter the pupil of your eye. And then some of these photons will actually illuminate light-sensitive cells at the backs of your retina. These are photoreceptors in your retina. And this will trigger a series of events within several cell layers in your retina that ultimately will trigger action potentials or electrical spikes to be transmitted down your optic nerves toward your brain for further processing. And every visual experience that you're having right now, every visual experience you've ever had, is a consequence of these action potentials streaming down your optic nerves toward your brain. That's what vision is based on. So one of the first stopping point for signals coming in from the optic nerve, the first cortical processing stage is in the primary visual cortex. This is in the occipital lobe at the back of your head, in the back of your brains. And there's a hierarchical network of several brain regions which process basic sensory features, basic visual features of the visual image. These are features like edges, brightness and contrast, color, texture, position, direction of motion, all of the kind of elementary sensory features that, if you were designing your own vision system, you would probably write programs to extract those features as well, right? So I call this the feature representation stage, and it mostly happens in this network of visual cortical areas in the occipital lobe of the brain. By the way, we know all of this from decades of studies, mostly from investigators going in and recording from individual brain cells. By the way, we each have something somewhere between 80 and 100 billion neurons in our brains, each of which makes somewhere between 10 and 100 or maybe even 1,000 connections with other neurons. So it's a really large-scale problem. But the way that this information's been gathered is by going in with individual electrodes and recording signals in individual brain cells in these brain areas. Okay, so information about the visual scene or visual features is first represented in visual cortex and then projects to a network of higher order areas which processes those sensory features in order to recognize the meaning or the identity of the things that we're seeing. And I'll call this an image recognition stage, where you'll see representations of object identity, object categories, and this takes into account the function or the meaning of the things that we see, right? So this depends critically on our past experience and critically on memory, not just what the thing looks like, but what it actually means in terms of the situation that you find yourself in. And so after you've recognized an image, the brain needs to then determine the course of action. What are we going to do? This decision would depend on factors like the current behavioral context, right? What situation are you in? The goals that you might have at the moment. Are you thirsty? Will you reach out and grab that coffee cup or not? Takes into account prior stimuli and events, expected consequences of different kinds of actions you might take, and your level of motivation. And then finally, there's the response itself. And this response is, you might be reaching out, grabbing the coffee cup and taking a sip. And we know that this action, this reaching action, requires commands sent from the motor cortical areas of your brain down your spinal cord to activate muscles in just the right way so you can reach out, grab the coffee cup, and take a sip. It took me a few minutes to describe this whole cascading process, which is, of course, greatly oversimplified. But in reality, this happens very fast, right? In a third of a second or something like that, you can decide your course of action when presented with some new object. So how do we study this process in the laboratory? Well, we train monkeys to play video games. And this is a depiction, this is a drawing of a monkey sitting in a custom-made monkey chair where the animals are sitting in front of a computer screen where we can project images onto the screen, and we can train the monkeys because they're highly intelligent. And by the way, their visual systems are organized very similarly to our own, anatomically and in terms of their ability to make fine discriminations or even to learn about the identities of objects. They can learn this as well. So we can train them to perform different kinds of tasks. We can train them to report to us whether an image matches some target image, or we can teach them different categories of objects. They can learn all kinds of things. So we can present images up on the screen. They might have their hand on a lever or something like this. And we can train them to pull the lever when they see a particular category of object, for example. Meanwhile, we can actually record signals directly from the brain, not gross measurements of average neural activity over some large area, but actually from individual brain cells, right? Targeting individual neurons, one of those in the monkey brain, maybe 10 billion neurons, we'll record from one at a time, except we can now record from large populations of these neurons using sort of larger scale multi-electrode arrays that can be implanted directly in the brain, giving us direct access to these signals. We're recording action potentials. These are spikes that are generated by neurons, which then propagate to their target, downstream target neurons. And these electrical spikes are the basis, the information representing basic unit by which the brain transmits information from one neuron to the next, and really from one population of neurons to the next. So it's sort of a massive decoding problem that we have as neuroscientists, because we wind up with large populations of neurons simultaneously recorded in these monkeys that are actively playing these games that we've taught them. And our goal is to try to decode what it is that the monkeys are thinking about, what decision are they going to make, and what is the formative information encoding in the brain that allows these cognitive processes to actually unfold. So the specific question I'll tell you about today, just one example of a project that we've had going on in the lab for quite a while, is how does the brain learn visual categories? Basically, how does the brain learn about the meaning of the things that we, or in this case, these monkeys are seeing? And what are the neural computations underlying the ability to make categorical decisions? Here's a side view of a rhesus monkey brain. The occipital lobe of the brain where visual features are represented is over there on your right. We've been focusing on a network of brain areas in a region of the brain called the parietal cortex. It's the parietal lobe is I think a really good candidate for mediating cognitive functions, especially decision making, because it's sort of intermediately positioned in between sensory processing and motor processing. Right, the parietal cortex gets inputs from the visual cortex, providing representations of the incoming visual sensory stimuli. And it's also interconnected with motor networks that actually, control the animal's actions. So and it's also interconnected actually with cognitive networks that are involved in representing rules and memories and things like this. So it's kind of ideally positioned to mediate flexible decisions based on incoming sensory stimuli. I'm labeling a couple of areas here. One of them, LIP, the lateral interparietal area, is an area kind of at the center of the posterior parietal cortex, which is especially important for visually guided actions. And this area, MT, is actually a higher order visual motion processing area it's one of those, visual feature representing areas that I talked about earlier. In this case, MT is really important for representing the direction of motion that is being presented in an image. So because of the particular circuit that we're studying, we train monkeys to make decisions about visual motion. So what we'll do is we'll show an animal visual motion patterns like this one here. So this pattern of motion moving upwards. Well, you can imagine we could present any different, of a wide range of different motion directions depicted by these 12 arrows here, 12 directions of motion. But what we teach the animals over the course of many weeks or even a few months of daily training sessions is we teach the animals that these directions of motion actually belong in two categories. There's category 1 and category 2. We invented these categories. They're defined by this category boundary that we made up. It's this green diagonal line that you see here, which divides 360 degrees of motion directions into these two learned categories. And so the animals weren't born knowing about these categories, they're learned, but they're very, very important because we query the animals using a categorization task, and they're motivated to perform the task because they love apple juice. And every time they get a trial correct, they get a drop of apple juice. The task is a delayed matching task. So the way it works, the animal's presented with one of these motion stimuli on the screen. So you see the sample stimulus, it's a motion pattern. The animal has to decide, was that category 1 or category 2? The animals need to remember the category they'd just seen during that delay period. And then a second stimulus is shown, a test stimulus. The test can be either the same category or a different category from the sample. And the animals need to decide, was it a match or was it a non-match? And they need to report that decision to us, usually by pushing a button or releasing a lever with their hand. It's a complex task. Why is it so complex? Well, we're working in an area of the brain that is, we know now is critically involved for the decision process. But we want to be able to tell whether this part of the brain is more involved in processing the sensory stimulus, the motion direction or motion category, or is it more involved in actually generating motor responses? And this is a task that allows us to separately examine those two factors. This task requires the animals to categorize that first stimulus, the sample stimulus, keep that information in short-term memory, and then compare the test with the remembered sample. Okay, so I'm just going to show you a couple examples of of what individual neurons look like in the monkey cerebral cortex while they're awake and performing this task. This is one example neuron. This is from the area MT, which is this area that's closer to the visual input, which we actually know from past work is critically important for processing visual motion. The time during the trial is shown on the x-axis. The firing rate of this neuron, the activity level in spikes per second, action potentials per second, is shown on the y-axis. And what you see is neurons like this one show a very strong representation of exactly what direction of motion the animal is looking at. The neuron fires the most if the stimulus was moving at 240 degrees, but fired less for any other direction of motion. And every neuron that we recorded from in area MT had a different preferred direction of motion, but we didn't see any evidence that the neurons were categorizing the stimulus, just that they were encoding what the stimulus looked like, what direction of motion it was moving in. But then when we looked downstream at the next processing stage in LIP, which gets input from MT, by the way, we found most neurons encoded the category membership of those motion directions, not the physical features of the stimulus. And you can see that because this neuron responds strongly to all 3 directions of motion in one category with a high level of activity, and it responds weakly to all 3 directions that were in the opposite category with this lower level of activity. So even though the animals are able to tell apart the different directions of motion, they've learned this rule through daily training sessions to tell them that they need to treat some of these directions one way, other directions the other way, and this has reorganized the representations in this brain region in the posterior parietal cortex. So the question then that we're really interested is can we explain how sensory representations in this upstream sensory processing area, MT, are converted into these learning-dependent cognitive representations in area LIP? And we want a real description of this. We want to understand a from a circuit standpoint, we want to be able to generate a wiring diagram, that shows us how is it that signals are converted from a sensory format to a cognitive format. It's a hard problem. It's especially a hard problem in an awake animal, in which you cannot completely record from the whole nervous system, from the whole brain. We can only conduct these sparse samples of populations of neurons. We also don't precisely know the anatomical wiring diagram. So this is actually where we've turned to artificial intelligence, AI approaches to train artificial neural networks. Like this is a depiction of a recurrent neural network here, to train to actually train these networks on very similar behavioral tasks that we teach the animals. And then we can, through analysis, reverse engineer the artificial neural networks to understand how is it that they're actually solving the task. So, these recurrent neural networks, like the one I'm showing here, this is the recurrent layer of this network is 100 recurrently connected neurons. And wherever possible, we try to use artificial neural networks that are biologically inspired. So we take inspiration from our knowledge of the way real neural networks are wired up and incorporate them in these artificial networks. For example, we have excitatory units and inhibitory units that follow some basic wiring rules that we know about from neurobiology. So the way this happens is basically these networks are trained through backpropagation through time to perform the very same task that I described to you earlier, this delayed matching to category task with visual motion patterns. The inputs to the network are from units which are visually motion direction selective. So they're meant to mimic the kinds of sensory inputs that are provided to an area like the posterior parietal cortex that we've seen in our experimental work. Then the outputs of the network indicate whether this test stimulus is a match or a non-match to that first stimulus, which is exactly the demands that the monkeys have on the task. The monkeys have to indicate whether that test stimulus is a match or non-match, and the target for training these networks is the same thing. So perhaps not surprisingly, I mean, these are very powerful networks, right? And this is an extremely powerful training approach, right? Backpropagation through time. Not surprisingly, the models can learn to perform the task with very high accuracy that is similar to the, to the 90% or greater accuracy that the animals can achieve. But keep in mind that this means that the networks are able to categorize that first stimulus, keep it in memory during this memory delay period, and successfully perform this comparison to indicate its response. But where things got really interesting is when we started analyzing the patterns of activity that we saw in the artificial networks that had been trained to perform the task. So here, after training is completed, we can go in and run the network on example trials and look at the activity patterns. So what I'm showing you here, these are 4 examples of neurons up in the top. The top row shows 4 examples of neurons from the monkey brain. 2 neurons are in the posterior parietal cortex, the ones on the left. 2 neurons are from the prefrontal cortex of the non-human primate brain. And these 4 neurons each show this kind of category selective neural encoding, like what I showed you earlier. Each of these neurons shows stronger activity for directions in one category or the other. Some of the neurons like the red category better, some neurons like the blue category better. But notice that there's also some heterogeneity in the dynamics of activity among these neurons. Some of them are very active right when the stimulus turns on, right when the visual stimulus is in view. That's at time 0. Others show this kind of ramping activity during the delay period. That's when the animal needs to keep information in short-term memory. Well, what really got me very excited, I was actually on the thesis committee for this student, Jia Chaiseng Mongkan. Through this long-term collaboration we have, my colleague Xiaojing Wang, she showed this slide or something very much like it in a thesis committee meeting. And it was a real light bulb moment for me, because these are 4 units from the artificial neural network that was trained on the same behavioral task as the monkeys. Now, we're not training the network to mimic the neural activity that we see in the experiments. We trained the network on the behavior, and then went in and to look at the patterns of activity. And whoops, how do we go backwards? Here we go. And what was remarkable is that we saw patterns of activity in the artificial neural network that you could have snuck them into some of our neural data sets from our experiments we might not have noticed. So this suggested to me that perhaps the artificial neural networks are finding solutions to this task that have something in common with mechanisms that are at play in the primate brains. This is very exciting, right? Because this means that this gives us a test bed for testing out ideas and hypotheses that we might have about how these decisions are made in the primate brain, but without having to do generation after generation of experiments in order to test out all these hypotheses. And it can help us, choose our experimental directions with more thought and maybe more carefully. And it's also just fun. It's given me a whole new way of thinking about other kinds of scientific things that we've been approaching. Okay, so although it's impressive that artificial neural networks can learn to perform cognitively demanding tasks and that they show some similarities in their activity patterns, as in the real brain, artificial neural networks suffer from some major limitations. I mean, we've heard a lot about successes today, but they're also highly limited, right? And one of the ways in which they're limited is that artificial neural networks, once they're trained, will tend to forget previously learned knowledge if they're retrained on something new, right? This is called catastrophic forgetting. And as the name would imply, this is this is typically devastating, right? So if we train a network on one of our categorization tasks, and then we retrain the network, we turn plasticity back on, retrain the network on a new category rule, the first category rule disappears, right? Unless we take very special steps to try to preserve that initial representation. So this basically occurs when learning a new task erases knowledge of previous tasks, and without strategies for maintaining past knowledge, artificial networks typically forget their past knowledge while learning something new. So one of the directions in my lab has actually, this is sort of an example of neuroscience guiding some new approaches to AI. We came up with a novel approach called context-dependent gating, inspired by the way that the primate brain can switch between tasks and learn multiple tasks without forgetting previous ones. And I'll tell you just briefly about this now. So there have been, this is a really kind of hot topic in artificial neural networks. Every year at machine learning conferences, there are many papers on this topic. And many of the sort of leading approaches in recent years have relied on stabilizing the synaptic connections in networks that were deemed to be important for a particular task. And so, one of these are sort of two of the leading methods for this: elastic weight consolidation and synaptic intelligence. But the basic idea is through ongoing analysis of the network weights, is to identify those weights that are key for a particular task and fix them during learning a new task. And this helps this catastrophic forgetting problem. However, it doesn't help enough. And it also is not very biologically realistic, because how does the brain know which synapses to stabilize and which should be plastic, right? There's some built-in mechanism that does not rely on post hoc analysis to allow this to happen. It's also computationally expensive. So our approach to this is inspired by our knowledge of how rule and task and context information in the frontal lobes of the brain is projected back to sensory motor networks in earlier parts of the brain that are governing visually guided actions. And the idea is to use these top-down connections to actually gate the activity of artificial neural networks in order to select which neural ensembles will be recruited to encode each new task or each new memory. And the way that we do this in practice is we have this top-down gating input, which sets to zero activity of 80% of the network when learning each new task. And this top-down rule-related input is also present when the networks need to then perform those tasks again later. And what we find is that this goes a long way toward alleviating forgetting in artificial neural networks, and also adds almost no computational overhead to the process. And so one way in which we've tested this is by training networks on the same kinds of tasks that I told you about before, but a whole long list of varieties of these tasks in which the network is presented with different visual stimuli and different task demands. Sometimes the network might need to attend to one stimulus or the other, or it might need to categorize a stimulus one way or another. But these are all of these different tasks that have different demands that are in conflict with one another. And we train an artificial neural network, a single network, in series to learn each of these tasks one after another, allowing us to ask, how well can a network maintain the ability to perform each of these tasks through this sequential training procedure? Only using synaptic stabilization, so these previous methods that had been developed, we see that that networks are able to maintain well above chance performance on these tasks with a mean accuracy of about 80% correct. Not bad, but also quite a bit of room for improvement. When we add on top of this our context-dependent gating approach, we see that this single network can now perform this large set of tasks with near perfect accuracy. Okay, so that's promising. We also find that it generalizes to a number of network architectures, including both recurrent neural networks, also feed-forward neural networks, and can even be used to alleviate forgetting in more complex contexts. So in this case, we trained artificial neural networks to perform Atari games through reinforcement learning. And in this case, the network was trained first to learn Pong, and then afterwards to learn to play Breakout. And what you're seeing here is just an example of performance of the network on both tasks, but after learning to play Breakout, right? So there was not like additional practice going back and training on Pong or something. So the network maintained the ability to perform the first game while after being trained to perform the second game. So what's exciting about this is it's a method for potentially maximizing or enhancing the representational capacity of a single neural network, using it to store much more information, packing more tasks and more memories into a single network than you might otherwise be able to without taking this approach. So we're extending this work in a number of different ways. We are one thing that we're doing is, and we're presenting this next month at the ICLR conference, a more learning-dependent version of context-dependent gating. Now, rather than those top-down connections providing a gating signal that is kind of hardwired by us, the experimenters, instead, these gating patterns are actually learned through training. So this makes it kind of much more flexible and potentially can help optimize performance. And a nice thing about this is that the relationship between different tasks is actually reflected in similar gating patterns being used for related memories or related tasks. So this is really helpful for linking memories that might be related with one another, or for recalling related memories when being presented with a cue. The other way that this is affecting the work in the group is it's motivating us to actually do new experiments, right? So this is generating hypotheses that we're interested in pursuing now by going back and doing new experiments to actually train animals on multiple tasks in sequence to try to identify what's changing in neural circuits as animals are learning multiple tasks in a single session. And this is something that we're working on actually right now in my laboratory. So just to conclude, brain-inspired context-dependent gating during learning can help alleviate forgetting for artificial networks that are trained on sequential tasks. And forgetting is alleviated by deploying partially overlapping neural ensembles for each new task, and by minimizing overwriting of important connections for previous tasks. And maybe just the broader conclusion is that this connection between science and AI is really bidirectional. I think there are many examples of that. Of course, not just from neuroscience. Neuroscience is maybe an especially interesting case, given that biological brains can efficiently solve many of the problems that are kind of currently very daunting for AI. And so asking neuroscience questions in artificial neural networks reveals plausible solutions to tasks, motivating new experiments, analyses, and hypotheses. And then adding brain-inspired features in an AI context can advance the capabilities of artificial networks, such as learning without forgetting, task switching, and continual learning. And so I'll just end there and thank everyone, in my lab that's made all of this possible. And, doing this work at UChicago is a real privilege, and it's really the right environment to do this kind of work. So very grateful for the chance to do so. Thank you.
Hey, it's a very trivial question, but in the very beginning of your talk, when you talked about the first stage, the MT area being like general classification of direction, and then the LIP area said, okay, now I'm going to turn this into a category. And you said that was the case, and it looked like the case. But did you take monkeys that weren't trained in categorization and measure their MT area and get the same result?
Yes. So yeah, that's a great question. And so yeah, MT is one of the reason we chose to study motion in this experiment in the first place is because there had been decades of prior work on MT. So it was sort of the place to start where we had the best understanding of representations in MT. And we've actually gone back and recorded more in just the way you've suggested. So in the adult, anyway, I mean, it's probably a whole different story during development before, visual critical periods, close up. But in the adult, MT seems unable to change its representation of visual motion. That's kind of true of the entire early visual system. That makes sense, right? If you, learn about, I don't know, some new image or new object, like you learn what the new iPhone looks like. You don't want that to change the way you see everything else, right? We want to have a stable kind of representation of the sensory world, but then plasticity in downstream areas that can store new kind of contextual or memory-like information. Yes, completely. Yeah, there's been a whole series of work of studies on that. In fact, they, not monkeys, but they've there were some studies where they raised cats in a stroboscopic environment. So with that, so their whole upbringing, they saw, only little brief snapshots, flashes of light, flash, 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 flash. So they saw lots of static images, but never saw smooth motion. And then experimenters went in and recorded from MT, and representations were completely alien and disrupted. So yeah, our ability to even see motion or see color or see at all depends on having, having visual experience early in life.
Yeah, that was a fabulous talk. It made me muse on something we were talking about at my table at lunch, which had to do with people making physical measurements of very tiny particles or in outer space and being overwhelmed with the data set, so overwhelmed that to actually make them tractable, they had to use AI to help to filter them. And I was thinking about this when you were talking, we're focusing on a circuit that creates a memory through one input, but the truth is that with the sensory system, there's an overwhelming number of potential inputs. So does the natural brain use some kind of filtering that is akin to something that AI would solve for, to know what things to actually pay attention to form these memories? How does it filter out the data? Are there analogies to this challenge that we're having in other large data sets?
Yeah, well, that's a great question. I mean, how much time do you have? Because there's so many interesting angles to that. But one of them, you mentioned attention, right? So, there's this myth, right, that people like to talk about. It's like, well, you only use 20% of your brain or something, right? Not true, right? But where does this come from? Why do people say this? I think this is a distortion of the phenomenon of visual of attention, which is that at any one time, your processing resources, your decisions are mostly based on a particular sensory input, or some particular memory or some subset of the information that's potentially represented by your whole neural network. And attention, whether something that's volitional and active, like keeping in mind a particular location that you're expecting something to happen, that's one way that extraneous information is filtered out, by basically ignoring anything happening outside of that realm. But there's also attention that can be cued by the external world. A bright flash, a loud noise in a particular location will reflexively attract your attention, right? And this is sort of in the brain how processing resources are deployed toward what is important at the moment. But that's sort of different than part of the question you asked, which was the scale of the scale of representations, the number of different areas that can represent a stimulus, and how that information is routed. It depends on what task is currently relevant. If you know you're playing tennis, then there's a certain task set that is loaded up that is causing your sensory motor system to work in a certain way that might be very different than when you're skiing, right? So there are these, but when I'm faced with an AI type question or an AI problem, I fall back on these kinds of ideas that I've learned about from neuroscience and cognitive neuroscience. And it gives us just our own sort of angle on thinking about how an AI network might solve a problem. Thank you.
So these are some fascinating analogies between phenomena that you can observe in the animal brains and animal nervous systems and artificial neural networks. But my understanding is that one fundamental difference between the two is the way that they are trained. So there's no backpropagation, or as far as I understand, no clear evidence of backpropagation, not in the same sense. So do you think that we're missing something about the electrophysiology of the brain, of how it actually operates? Or is it that that networks that are trained in fundamentally different ways still exhibit the same metaphenomena?
Yeah, it's a great question. So there's sort of two parts to this. One thing that is generally missing from AI models that I think all the talks we've heard about so far today, everything that I've spoken about, are all using artificial networks based on rate-based units, right? Not spiking units that are spiking in the sense that like neurons in the brain fire these discrete spikes. And so that is a really important distinction, because some of the key learning mechanisms that we know exist in the brain depend precisely on the timing of spikes relative to one another in different neurons. Two neurons will become more strongly wired together if they are co-activated, but with the sequence where neuron 1 fires first, then neuron 2, following causality. Those kinds of learning mechanisms are very difficult to approximate in rate-based artificial networks. Now, spiking networks exist too in AI, and they're really useful. They're actually going to be really useful in hardware implementations, right? All of the sort of neuromorphic chips, those tend to be spiking based because, ones and zeros are very cheap to represent compared to, graded activity levels. So I guess what I would say is that, yes, we are using learning approaches that are not, as far as we know, very biologically realistic, but it gets these networks into a state where they can perform this task that we're interested in. So it kind of in the fully learned state, we have a nice system to analyze that is satisfying. But what we're not trying to do quite yet is studying the, trial-to-trial learning process, because backprop doesn't really lend itself to that. Thanks.
Thank you. I have two questions. One is I noticed in your presentation, you use the frequency of the neural signal to measure the intensity. What about the what they call the power, the amplitude of the signal? Is that something doesn't matter, or this is just for
these particular analyses? What really matters is frequency or rate. So the variables of interest, to the best of our knowledge, are encoded by firing rate of particular neurons in small time windows. A spike is a spike, for the most part, in a particular neuron. Every spike will look the same, and so it's kind of an all or one phenomenon. So the shape of that action potential doesn't matter. What really matters is the frequency with which they're produced.
Okay. The second question is in your what they call the gating method,
Mhm.
like you have a like forget like a ratio. I remember it's 80%.
Yes.
Is that the number you kind of experimented to be the best, and what kind of criteria using to choose that?
Yeah, that's a good question. And that's actually something that's become more flexible in our learned version of this, where we relax that constraint. That was a starting point for us. And you know, that particular algorithm works over a pretty wide range of those values, but we thought that was a reasonable starting point. But yeah, we could talk about it more.
So that
That's-
basically
yeah.
what I mean is that
Yeah.
number is relatively robotic. No, the kind of stable. Like if you
Yeah.
change that number a little bit, it should not be sensitive to your result.
Right. That's right. Yeah.
Okay. Okay. Thank you.